first of which is, I encourage those of you who are available tomorrow at 12:30, come to the first of our spring science talks. I'll be hosting Dr. Platt from Georgia Tech and Emory. So I've been doing a little research on introducing him today. It looks like it's going to be really, really cool. This is our science talk is an interdisciplinary seminar, so the idea is that it's a college-wide talk that we touch on a lot of facets of chemistry, biology, computation, etc. So for an hour tomorrow, you can come see what I hope will be a really cool talk about proteolytic activity in disease and predicting and modeling disease potential. If you'd like to meet with Dr. Platt, I can arrange it to see me after class. And I would point out, those of you who want to go to grad school could do a lot worse. If you just look at his research lab website, he's doing everything from translational research in sickle cell disease to integrative networks in cellular systems. So he's very interdisciplinary. I suspect, I've talked to him on the phone a couple of times and never met him in person. I suspect it'll be very interesting at the least. It can open up for you a little view into what's going on. The biomedical engineering department he's in is shared by Emory and Georgia Tech, so it can be a good entree to grad school. And one of the important things, he's got a $2.25 million grant. So you can also probably pay for it. That far eclipses me, by the way. I'm very jealous. It's all about who dies with the most great dollars. I hope you enjoyed your vacation. It was more like a prison sentence for me. I had 6 straight days of single parenting. My wife was working in the hospital, so she walked to the hospital and then went on the girls' weekend trip. So I found excuses with my 3 children to avoid prepping virology lectures. About Tuesday at about 12:06, I'm like, yeah, my chance of having to teach tomorrow are pretty minimal. So I just wanted to take a second, now that I'm child-free, to let you know how I'm going to do this. There was a little post on D2Up in case you missed it. We were supposed to do Chapter 6, and the exam is a week from Wednesday, so what I'm going to attempt to do is squeeze 6 and 7, and if we get that far, a little bit of 8 into today. On Wednesday will be the Chapter 8 quiz if you want to do it. And we will talk about, include the discussion not of the paper I was going to do last week, which I've scratched, but rather this week's paper, which can be found, the link to which can be found on the D2L site. So it's a PNAS, which is the Proceedings of the National Academy of Sciences. Used to be known as Publish Nearly Anything Suckers, but has gotten much more exclusive in recent days. So it's a short paper. I want to start getting you guys at least looking at primary literature, so if we read that prior to Wednesday, we'll have a better discussion. Relative to next week, what I attempted to do last week, we'll do this week and then next week, is pick something that's discussed on the This Week in Virology podcast. And I couldn't decide which one to do for next week, so I'd like just a quick show of hands, half-baked opinions. So the TWIV that went up yesterday has 2 papers that are at least of nominal interest to me, one of which is H5N1 influenza transmission, which is more of an experimental paper versus a current opinion paper, so sort of an editorial commentary on antiviral options for biodefense. So show of hands, who would rather do something about flu? And who would rather do something about biodefense? Flu? Biodefense. All right, flu carries it. So I have to request these through interlibrary loan, but it'll be this one from virus research. And I'll also post the one for biodefense, since I know those of you who will be disappointed by having just lost that vote will want to read it anyway. Okay. So as promised with the mnemonic device, it's so bad I can't even remember what it was, ATGAY, attachment, entry, transcription, translation. genomic replication. Exit. Okay, so I guess we did A and E. We're going to do transcription, translation, and genomic replication today. And to start with, let's briefly talk about the central dogma. Generally, we should avoid dogma, but this was proposed by Francis Crick, who, of course, is one of the discoverers of the structure of DNA. That immediately, actually even before they had the structure of DNA, he proposed the central dogma. If you ever look at the paper that reports the structure of DNA, there's a little blurb in there, something about the structure of the molecule suggests a mode by which replication could occur. And so we have this is represented differently in your textbook. This is something I like to do. Protein. So the central dogma is genetic information flows from DNA to RNA to protein. Protein's what's doing the work of living. DNA is doing the information storage. And of course, this is replication. This is, transcription. This is translation. Let's take a moment to think about those two terms. Undoubtedly, you've seen them many times before. But what's the difference between transcription and translation? The words, not the biological processes.
Transcription is to write, translation is to read.
What are you writing when you're transcribing?
You're copying it over. You're just copying information in saved format.
Or you're writing down something someone is saying. You're not changing languages. You're not changing alphabets. You're just making a copy or changing formats. So you could go from spoken word to written word. It carries the same meaning. So you're changing the writing. Whereas translation, we're changing languages. So these terms are very fitting for the biological processes, right? DNA and RNA are effectively the same language. The codons there, DNA and RNA contain the same information, namely what we want, what amino acid residue we want when we get to the protein level. So incidentally, DNA also codes for its own copying, replication. That's chapter 7, by the way. There's an overlap with chapter 6. Now, the book mentions, goes to some pains, I'll go to greater pains to say the central thing you need to know about the central dogma is that it's wrong. DNA doesn't flow to RNA, doesn't flow to protein. As originally proposed, these arrows were one-way arrows. As the figure in your text shows, there is some flow in opposite directions, modified in 1970 or so, where we can have, for example, reverse transcription. So I'm representing the way I like to figure A. We have figure B, in that genetic information can flow from RNA to DNA. Well, that's not the end of the story. We won't touch on it in this course. But of course, protein can, in a way, encode additional copies of a protein. Does anybody have a guess as to what that might be?
Thing about the proteins that like make copies of themselves in like prion diseases.
Prions. Right. So,
Mad cow.
or as I prefer, bovine spongiform encephalitis. Prions, for example, they don't encode additional molecules, but one protein misfolded can cause another to misfold. So it's sort of a catalytic event to produce another molecule that does the same thing. So we have, and of course, we also learned we can have RNA to RNA replication as well. So there's a saying, I'm forgetting who said it now, all models are wrong, some models are useful. So the central dogma as originally proposed and as amended for your textbook and is just amended by others, and in my schematic here, me, make the model probably still wrong, more useful in terms of thinking the flow of genetic information is more than one direction. Replication does not only occur at the DNA level, but also the other levels. But the overall point is true. DNA, storage material, that's where genetic information is most stable, most easily replicated, readily transcribed into a usable form. That usable form is readily translated into protein. So, meh, good enough. So in order to transcribe the information from the RNA copy of a viral genome, we need various things, and it depends on what type of RNA or virus we are. So in chapter 6, your text goes through that there are 7, depending on how you're counting and what bar you're having to fight about how many there are, 6, 7, or 8 classes of viruses that break down to double-stranded DNA, single-stranded DNA, yada yada. And so you'll note if we have an RNA virus that is the template strand. And of course, we have 4 types of viruses. Single-stranded or double-stranded of either one of the 2 classes of nucleic acid. If they are the single-stranded forms, they can either be the plus or minus strand. That minus and plus is with respect to mRNA. So, if it's the mRNA form, so class 4 virus, its genome is the messenger RNA, so it can be directly translated. We saw an example of that with rabies, but West Nile is a plus-stranded RNA virus. A minus strand can serve, so a plus strand cannot be replicated directly to the mRNA because it's complementary to the minus strand. So as we'll see, when transcription occurs, it occurs from the minus form, which is the template, to the plus form, which is the molecule that will be translated. So let's talk for a moment about transcription. To do that, I want to bring up the lac operon. Has anybody not ever experienced the lac operon? Who, raise your hand if you have not gone over the lac operon in a class. You've all taken genetics? There's one. Been on, okay. Who is uncomfortable at remembering how the lac operon works? Thank you, brave souls. All right, so some of you who are not brave souls, because you either already know it or don't want to admit to it, what's the lac operon? You guys are exempt from asking this question.
It's a bacterial nucleotide sequence that operates in the presence of lactose.
Yes.
The
Okay.
production of genes.
So let's back up. What's an operon?
Something that changes shape, or this one changes shape.
No, that's a
right?
doppelganger.
Don't you have multiple genes under one promoter?
No, what's a shapeshifter? Like Mystique from the X-Men.
Squirrel.
No, chameleon?
Chameleon, no.
Chameleons change.
Okay, yeah, it's a shapeshifter. Doesn't change shape. So squirrel, thank you. Back to operon. What was an operon? Yeah.
Multiple genes under one promoter.
Group of genes under, yes, let's take that as a, we haven't introduced promoter yet. Commonly regulated. Okay, the lac operon is a group of genes in bacteria. E. coli is where it's been worked out best of all, that control the genes that are responsible for lactose metabolism. Why do we need to control lactose metabolism if we're, put yourself in the mindset of E. coli for a moment?
You don't want to express that gene unless you have to.
Why not?
Because it's a waste of energy.
Why?
Takes energy.
So yes, I'm sorry, I didn't give you enough positive reinforcement. That's exactly right. Because you know it's a waste of energy. Yes, that's exactly right. Why?
It has to release that operon in order to express that gene, right? That
You get to
takes
ramp up and cell machinery to bind
It
those
takes
substrates.
2 tryptophan molecules to get in there.
2 tryptophan.
Or not tryptophan, but
Now you've gone off words.
it has to have the lactose molecules
Right.
bind to it to change shape, and that has to be transported into the cell, into the nucleus. Right?
Yes, let me rephrase that as a professional. E. coli doesn't want to metabolize lactose if it can avoid it, because it takes biosynthetic energy to make the proteins to metabolize the lactose. Lactose is a disaccharide consisting of 2 monosaccharides, not tryptophans, that need to be hydrolyzed to, that has the glycosidic bond hydrolyzed to monosaccharides, which can be transmuted into glucose, which is the
Preferred energy source.
preferred energy. And why is it the preferred energy source?
For ATP production. It's
Yes. Where do you make ATP? E. coli don't have mitochondria.
That's right. That's true.
The ATP and
Glycolysis.
Yeah, glycolysis is right. It's the starting point for glycolysis, which is where we begin to make ATP. So, if you have glucose available, let's say we're a lab strain of E. coli and we're growing in a rich medium, we don't want to turn on the lactose metabolizing proteins or genes to make the proteins because we have plenty of glucose. So it's only in the case, and it gets a lot more complicated than I'll get into today, where if lactose is present and glucose isn't, we not only turn on the genes, but also up. So there's on, off, and up, down. And so you can imagine the lac operon, not imagine, this is what it is, but structurally, as a promoter, an operator, and 3 genes. So bacteria, as you'll see in a moment, can make polycistronic mRNAs. That means one mRNA can have 3, or it can have more than one polypeptide associated with it. In this case, there are 3 translation start-stop sites coding for 3 separate polypeptides, one of which is beta-galactosidase that cleaves the lactose in 2. The other is lacpermease, that's the channel protein that lets it in the cell. And the other one, oddly enough, at least the last time I looked, is to this day functional unknown. Lac, which I think permease is A and lac Y. Don't quote me on that. But I'm bringing this up to illustrate a couple of concepts, one of which is promoter. So define promoter for me. Somebody who organizes concerts and make sure that
Really regulate how the gene is expressed.
What's
what is a promoter? Sorry, that's what I just asked, is it? A promoter
It's
is
a
a,
recognition
It's a
site, like a
sequence of-
transcription factor.
not just, yeah,
It binds to it.
almost. It's the binding site for RNA polymerase. So, where the polymerase that's going to make the RNA transcript binds to the DNA. The operator is also a binding site. Either protein-nucleic interaction, we're talking about RNA, all. For our purposes, all RNA polymerases are the same. This is the binding site for a repressor molecule. So I have an animation showing you how this is going to work. We'll view several animations today. But the concepts I want you getting are the same ones that are going to come true, that are going to hold true for our virus transcription. We have a promoter, the site to which the polymerase binds. We're going to have not operators, but enhancers that can turn transcription up. The operator serves as a binding site for turning things down/off. We'll work the reverse with the intermolecular interactions are the same. So I'm going to show you a couple of videos in this class, or animations today. They're all from an NSF-funded website called the Virtual Cell Animation Collection. Oddly enough, run out of, I believe, the University of North Dakota, of all places. I think we'll 3 of them today. This is the lac opera. I want you remembering promoters and enhancers, and I'm using an example from bacteria because 1, you've seen it already, and 2, I knew where the animation was.
E. coli lac opera is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon, beta-galactosidase and permalase. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
I've written down my notes here. It's more complicated. I'm like, why is this more complicated? The lac operon is more complicated than the animation you just saw. There are other elements for enhancing and inhibiting transcription. But again, the basics hold true. Your promoter is the binding site for the polymerase. The operator affects not binding, but whether transcription occurs. So we're going to talk not about operators, but promoters and enhancers. So promoter, binding site for polymerase, enhancer, the binding site for transcription factor. So E. coli needs the efficiency brought about by having that regulatory scheme in order to survive in an environment where it needs to not be wasting synthetic energy. Viruses might need something else in their promoter. So what are you going to guess a viral promoter is like compared to a bacterial promoter or your garden variety eukaryotic promoter? What's going to be the overarching characteristic of a viral promoter? What do viruses need to do when they cause infection?
They need to hijack the cell's machinery.
Right, they're hijacking the cell's transcription, translation machinery. What's that going to mean relative to the promoter?
Promoter would be fit for the whatever polymerase it needs from its host.
So let's say, for example, we have a DNA virus that needs to go into the nucleus. We'll talk about that in a few minutes. Gets in there. There are a bunch of polymerases around. The host has defenses, right? It's going to try to chop up DNA, do various other things to it, rewrite it, etc. So what's a promoter going to, viral promoter is going to be?
Similar. It'll be similar to the host.
Similar. Well, okay, let's go back to what we, so yes, actually, that's right. It's going to have a ta-ta box, it's going to have those characteristics your textbook mentions. Just not getting there, my fault. It's going to be really, really strong. You're getting there, it needs to hijack the machinery. So another way of saying that is it needs to outcompete eukaryotic promoters for those polymerases. Otherwise, it's going to lose out in terms of transcribing, making its proteins, assembling viruses, and exiting the cell. If let's say, a weak promoter, given weak enough promoter, the cell could virtually ignore it. It's turning on its own genes and transcribing its own mRNAs and making its own proteins. So a weak promoter wouldn't be very infectious, so that virus would be a bad virus. What does strong mean? It readily binds, so phrase that in terms of what we talked about last time. It has a, does it have a higher or lower KD?
Lower.
Yeah. Which means it has a higher affinity. So strong promoters, higher affinity for polymerases. That's my point. So, not coincidentally, by the way, when you're in the lab and you're doing tissue culture and you want to express a foreign protein in a mammalian cell, and then it gets, for whatever scientific purpose you have, very commonly plasmid vectors are used that have viral promoters. That's also true in bacteria, but SV40, which is simian virus something, number 40, is a very commonly used promoter because it's so strong. You can put your plasmid into virtually any cell and it gets transcribed like that because of the affinity of the promoter.
That's crazy that they developed that.
Crazy, yeah, sure.
I mean, it's cool.
Right. So, and the bacterial vectors are the same way. So you talk about the lac operon. The lac operon is the most frequently used bacterial expression system because you can put your gene of interest on a plasmid, put it in the bacteria, and then trick the bacteria into thinking you've given them lactose. You use a chemical called IPTG, stands for isopropyl-lactopyranoside. It's a mimetic of lactose, but the beta-galactosidase can't cleave it, so it can't get rid of it. So it's always on, and so the bacteria goes, holy crap, I need to metabolize lactose, but it's really making your protein instead of the lactose. Of course, so your textbook here is drawing a gene to be transcribed as DNA and then polymerase, and the promoter is here, or the promoter is here. an enhancer, which is a transcription factor, binds up here. So can somebody explain to me what's happening here vis-a-vis promoter enhancer, polymerase transcription factors, and whatever this is? Let's explain that. What does an enhancer do?
Increases transcription.
How?
Stabilizes the binding.
Right, yeah, oh, so they're enhancers, they're negative enhancers as well, but we're not talking about this. So it increases the rate of transcription of the gene. So if you're a virus, you've got a very strong promoter, you want to make a lot of copies of the mRNA because you want to get the hell out of Dodge before the cell dies or kills you. You have a strong promoter to turn on transcription. You have enhancers to turn up that strong promoter. How do they do that? With an enhancer. What's going on?
I guess, are they arranging the DNA to be more favorable to be transcribed?
Well, so there's another enzyme that unwinds the DNA for us.
Polymerase.
Yeah, so let's not worry about that right now. So no, it's not arranging the DNA in that many words. The enhancer is usually found 5', not always, to the gene, but enhancers close by are generally 5' to the gene. So you would see some arrangement like this in space. This is schematic. What's binding to the enhancer? It's a transcription factor. How does that transcription factor increase transcription? Asking nicely.
Upregulation.
So yeah, it's going to upregulate, but what does that mean? It means increased transcription. How does it increase transcription?
It just binds to the enhancer sequence.
Okay, how would that increase transcription?
It allows the RNA polymerase to bind more readily to the promoter
Is it a
region.
confirmation, like change?
Something's there, but
So there's a missing.
it binds to the, so a transcription factor binds to the enhancer, and you said what again? More readily.
More readily, or increases the affinity of the RNA polymerase to bind to that sequence, the promoter region.
Yes, how does it do that? The answer, by the way, is always binding. It's got an affinity
Is what it does.
for the RNA polymerase. So the answer I'm looking for, and I think you're basically saying this, is the enhancer binds the RNA polymerase, the promoter binds the RNA polymerase. We have two things with affinity bound to DNA that's going to increase the overall affinity for the RNA polymerase. So the point I'm trying to get to is promoters, enhancers, transcription factors all serve to recruit an RNA polymerase there. There's then a hydrolysis that starts transcription, leaving the promoter there again. So leaving the promoter open to finding another RNA polymerase. So a strong promoter is going to have a high affinity, an enhancer is going to have an affinity for the RNA polymerase. When the enhancer is bound near the promoter, you're essentially going to have A plus B goes to AB plus C goes to ABC, and ABC is going to be a very stable complex. So those affinities we talked about, we didn't get into kinetics as much as rates, work to the advantage of the virus in terms of turning on transcription of the RNA to the gene. So, let's watch another animation.
Our transcription animation provides an overview of the major steps involved in RNA production. In this animation, we will take a closer look at the events that prepare the DNA for transcription. mRNA and eventually proteins are produced when and where they are needed in an organism. Some genes, such as those for housekeeping proteins in processes like glycolysis, are expressed all of the time. These genes undergo what is known as constitutive transcription. Other proteins are only produced at specific times in certain cells. In these cells, genes undergo a process called regulated transcription. Transcription is regulated by proteins called transcription factors. These factors are proteins that are produced in the cytoplasm and eventually migrate into the nucleus, where they interact with DNA and activate transcription. These transcription factors only interact with specific genes, those genes whose transcription they control. Most eukaryotic species have over 1,000 transcription factors. Regulated transcription begins when a signal is received by the cell. This signal, often a protein, begins a signal transduction cascade that lets the cell know certain proteins are now needed. The most common activation event for a protein in a signal pathway is the addition of a phosphate group. That activated protein in turn interacts with another protein. This interaction leads to the phosphorylation of the next protein in the pathway. There is often a series of proteins that are activated by phosphorylation. At the end of this cascade, the final protein will be activated and then enter the nucleus. It must make this journey because transcription only occurs inside the nucleus. After entering through a nuclear pore, the protein interacts with a specific transcription factor responsible for activating mRNA production. As with the earlier proteins, the transcription factor is modified through phosphorylation. The activated transcription factor will next bind to an enhancer region. The enhancer is a region of DNA upstream of the transcription start site that binds a transcription factor. The transcription factor then binds to the DNA and moves to interact with the rest of the transcription protein complex, located at the transcription start site. When the formation of this complex is complete, transcription of the gene will begin. It is this sequence of events that ensures genes required for a specific tissue or only at a specific time are expressed appropriately.
I take a little bit of an exception here to the animation. Why might I? Think like McMurray for a minute. What's wrong with that?
They can be upstream or downstream.
Yeah, that's true, but that doesn't upset me.
DNA could be all wound up and the polymerase might not be able to get to it.
Yeah, it could be packaged in a chromosome, except it's a virus. And not this one, but yeah. Also not upsetting to me. What is the argument I just made about affinities? Yeah, pisses me off. No. It's showing you the RNA polymerase is already assembled on the promoter, and the transcription factor comes in and binds, and then everything happens as normal. It's this polymerase sits down on that promoter more frequently for having been turned up by this transcription factor. So you saw it was an insulin signaling cascade of phosphorylating things to the point where a transcription factor gets turned on as a result of the external signal. Viral entry into the cell can also turn on transcription factors that favor the virus. But it's this and this that result in this being bound more often. I think it's hard to show in an animation. They're just focusing on the basics. But as you can see, it's just like the lac operon, except it's different. So, on and up, transcription occurs via polymerization of a strand of RNA based on hydrogen bonding and catalysis of the hydrogen bonding of the nucleotides as they enter the polymerase, and then the catalysis of it's a phosphate transfer reaction. But for now, we make a RNA strand as a result of the template, so it uses the minus strand to make a copy of the plus strand, and we get an transcript or mRNA. In eukaryotes, of course, there are introns and exons. The introns must be removed to make the mature transcript, which codes for the protein. Viruses can also do this. That gives them the ability to do what? I just realized I was giving it away with my motion, so I stopped. What's the point of having introns and exons?
So you can alternatively splice DNA or splice
You can make different proteins from the same gene. Why is that an advantage to viruses in particular?
You can control which proteins are made.
Because they're small. They don't have much information. You pack more information in an mRNA that can be alternatively processed from a primary to several different secondaries. So this shows that different viruses transcribe in different ways. I just want you to hit the highlights here in that if you're an RNA virus, to make a new RNA molecule there's not a whole lot of thunder here. Right? What this tells you is pretty much everything. Anything can happen anywhere. It depends on the particular virus and the particular lifestyle of the host. The double-strand RNA can use a double-stranded RNA-dependent RNA polymerase to make an mRNA. So this is transcription that's occurring in the cytoplasm. Normally transcription occurs in the nucleus. So a minus-stranded or a single-stranded minus RNA molecule has to use a virus single-stranded RNA-dependent RNA polymerase. Why? Because the host doesn't have one. Right? If you're double-strand DNA, you can use the host RNA polymerase or the cell's DNA-dependent RNA polymerase. So independent, well, it's not independent. Different viruses do different strokes for different things. But the shorthand used in your text throughout will be which molecule RNA dependent, and they're all RNA polymerases here because it's transcription, so they're making RNA from that. Like eukaryotic messenger RNA, the RNA are processed. They're using the host, so it makes sense that they're going to have the similar processing to host mRNAs. For our purposes, that's a 5' cap and 3' polyadenylation. Both of these modifications enhance the stability of the mRNA. There are exonucleases, enzymes that hydrolyze RNA starting from an end, as opposed to an endonuclease that would start from the interior of a molecule, like a restriction enzyme cuts not at an end of the molecule. So if you have a cap, which is the first base at the 5' end has a methylated, or the 2' hydroxyl is replaced with a methyl group, and then the cap is on what would be the 5' phosphate, but is instead capped with the 5' cap. Thus, it's not a substrate for an exonuclease. There's a template-independent polymerase, so it doesn't need a DNA or RNA template. It just adds adenine at the end, so it's called the poly-A tail. Serves the same purpose. The longer the poly-A tail, the longer it takes exonucleases in the cell to break down an RNA. You remember back to my comments earlier in the semester, the purpose of RNA or RNA is unstable for a reason. That's because when you turn on a gene, you want to be able to turn it off. So the way you turn something off at the translational level is to destroy the transcript. So these enzymes in the cell exist to destroy the RNA after a while. That's, by the way, what this methylation is designed to prevent, is that prevents hydrolysis here, so thus it increases the stability that the cap provides, because we can't have the 2' induced hydrolysis that we can in the rest of the molecule. The cap also serves for an initiator of translation, as we'll see later. Most, but not all, viral RNAs are monocistronic. We have some examples of bicistronic mRNAs as well, which means one open reading frame per molecule. That is, in a mature mRNA, from virus garden variety codes for one protein. So here you see cap, untranslated regions 5', 3', we get to a start codon, we make one polypeptide from one mRNA. Except when we don't. Oops. Except when we don't. So you can have an mRNA that has two open, or you can have mRNAs with more open reading frames. Bicistronic is much more common, and you see that the open reading frames can either be overlapping, there's just a little bit of the reading frame that's there. One can be internal to another, and the leaky scanning here is, I haven't done translation yet, have I? A ribosome, instead of finding the start codon here, will miss it and find this start codon down here. So this is another way of embedding more genetic information in one mRNA molecule. You can code for two proteins, and due to essentially mistranslation, you can get more proteins. I never dealt with translation. So let's move on to translation. We have our mRNA, our start codon. By the way, I may ask you to do some translation on the test. I haven't decided. Oh, sorry, back up. I can't have to. I may ask you to do some translation on the test. If I do, I will give you the genetic code. So you don't have to know that AAA codes for. Take a guess. Wrong. I see. Fortune favors the bold. So I may ask you, but I'll give you the information, and I may play a trick with you on what's the open reading frame, something like that. But you need, if you can't do it, you need to be able to go back and look at your Bio 1 or 2107 book and say, here's the table, what does it mean? So if you need help with that, I'll be happy to help you outside of class. So, translation, of course, is going from mRNA to protein. That's done by the ribosome, which consists of two large subunits, the 40S and 60S. Does anybody know what the S stands for?
Svedberg.
What's a Svedberg?
It's more about the speed.
It's a unit of measurement that relates to how fast something goes down to the bottom of the tube in an ultrasonic. So it's not only mass, but shape. So 40 Svedbergs doesn't relate to a certain number of grams per mole. Svedberg is big, 40 is massive, 60 is even bigger. So two subunits that come together on the RNA, and what you see here is initiation of translation. That 5' cap serves as a binding site for initiation factors. The E stands for eukaryotic. So the virus is not producing its own translation factors. This is where it's using the cells. They bind to the N, they recruit the 40S subunit, which comes in with its own tRNA that has formulated methionine in it. And that 40S subunit will scan along, find the start codon, which is always AUG, except when it isn't. And there are a lot of exceptions to this, but for our purposes it will be AUG, at which point the 60S subunit comes in, and then translation begins. Let us review translation and animation. I like these animations.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the amino acid, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
I expect that's entirely a review for everyone in here. It's not necessary that the mRNA have a cap. If it doesn't have a cap, it's going to have a sequence that recruits the 40S subunit anyway. And that's called a ribosomal entry site. So it's a sequence of the nucleic acid or RNA that folds up into a structure that has an affinity for the subunit itself. The I is internal. So, post-translational modifications, we're now up to we have the polypeptide of our viral gene. We've transcribed, we've translated. The post-translational modifications, experiences we tour our viruses this semester. Glycosylation, this is a sugar moiety added to either serine, threonine, or asparagine, depending on the type of the carbohydrate additions. This most happens, occurs on viral I'm sorry, that's shorthand for carbohydrate. So you can see furrowed brows. Sugar. CXH2OX. This is going to occur on viral proteins that are parts of coats and envelopes, so it's pointing out. Glycosylation occurs in eukaryotes on proteins that are going to be either secreted from the cell or cell integral membrane proteins, that parts of which are outside the cell. So acylation is addition of a fatty acid moiety, so we have a long carbon chain that allows for membrane anchoring. Again, important in enveloped viruses. Phosphorylation, as you saw in the animation today, is the most common way to turn something on or off. This, of course, is addition of a phosphate group, and that's to turn things either on or off. Your text barely touches on it. It's less important for viruses than it is for real organisms, but we need to turn things on and off. When there is viral gene regulation, so many viruses will have early expression and late expression, where they'll make capsid structural proteins before making the proteins necessary for viral maturity. So phosphorylation does occur on viral proteins. And then proteolysis. So that's hydrolysis of peptide bonds. Many proteins need to be cleaved in order to be active. So we talked a little bit about blood coagulation earlier in the semester. Blood coagulation factors have to be cleaved in order to be active. So many viruses will make polyproteins, which are one mRNA transcribes more than one, or one polypeptide that when cleaved will form multiple proteins. So the best example of this is HIV protease, that for several of its structural proteins, it makes a polyprotein, and then it encodes a protease, HIV protease, that cleaves those polyproteins into the mature proteins. I just pulled one example from the literature here. HIV protease is a major target of therapeutics, and there are a number of drugs on the market now that are inhibitors of HIV protease. So this paper just shows one, it's a dimer, so there are two polypeptides of HIV protease. There's the active site where the cleavage occurs. It's got two things it calls flaps. They're in the mechanism of the enzyme. They move about 7 angstroms, which is a big movement, or if you're in England, angstroms, a big movement. And so this paper is showing, it does a lot of crystal structures, and shows that topranavir, not one I was terribly familiar with, binds to the flaps and keeps them from flapping. And that is the mechanism for inhibition. So it causes a rigidity in the protease that inhibits proteolysis. Why is HIV protease a good target for therapeutics?
Without it, the virus can't finish its replication.
Yeah, right? Polyprotein has to be cleaved. If you inhibit the enzyme, can't make new viruses, you've prevented the virus from replicating, you've saved the patient's life. So it's an excellent target because it stops HIV from proliferating. But why is it an even better target than the one I just thought of, I almost said, is also a good target. Other stuff the virus does. So let's think like pharmacologists for a minute.
Because then you don't have to treat it.
I mean, the other stuff is stuff the virus is using that's cells on machinery, so that could cause all sorts of bad side effects, where the HIV protease is specific to the virus and then made by the body.
Well, you just killed off 10 minutes of discussion. Yeah. So HIV protease is specific to the HIV polyprotein. So HIV protease represents a target that if you can hit it, doesn't mess with any normal cellular functions. So a good drug causes the effect you want it to and no side effects. Now, there's no such thing as a drug with no side effects, at least yet. But you can have a drug with fewer side effects. And one of the things, if you have a high affinity for HIV protease, it's an aspartic acid protease, so there are normal aspartic acid proteases. An active site inhibitor, which is a lot of other drugs that look like the substrate, but they're not, might bind to other proteases and cause problems. So this is a good idea because it's a flap to a protease that only HIV needs. The side effects are likely to be minimal because you're not going to be mucking with any normal biological process. So it's a good therapeutic because it's a good target, and it's a target that if the missed shots at the target are unlikely to cause damage. So it's one reason why it's a very good therapeutic. It's used in combination with other things like, well, I guess AZT is kind of over now, but there are other nucleotide mimetics that affect the reverse transcription. That was supposed to be the interesting aside so that you wake up. So, the class is transcription, translation, transport, so we're up to the transport part. Host-supported transcription occurs in the nucleus. In order to get translated, the mRNA has to leave the nucleus. The nuclear import and export signals, so that, well, the mRNA, there's the machinery for getting the mRNA out. Cytosolic translation of an mRNA, so a plus-stranded virus doesn't need to be transcribed in the nucleus, but that protein that it encodes might need to. So there are nuclear localization signals on a protein that can take that can result in viral proteins going into the nucleus and also out of the nucleus. The transcription occurs in the translation occurs in the cytoplasm. For membrane proteins, translation begins to occur, is arrested, the ribosome associates with the endoplasmic reticulum. So the endoplasmic reticulum these are membranous structures. I'm assuming everybody knows what an endoplasmic reticulum is. But that flow of the lipids and the proteins is generally from near nucleus to out. So we're going to have, for example, a viral membrane protein needs to be translated at the ER. The ER needs to transport that protein. Well, the protein is transported from the ER to the Golgi, from near Golgi to the far Golgi. Could be vesiculated, could be exposed to the cytoplasm. This is nothing other than the viral protein encodes its address localization the same way a cellular protein that would be displayed on the outside, for example, a receptor would as well. We can see viral proteins expressed on the surface. These are going to likely bud off into a new viral envelope. Epithelial cells are weird. They have sides, so there's sidedness. This is the apical membrane, the basolateral membrane, so this is exposed to, say, the lung. This would be non-epithelial cells. Different proteins translated in different ways can be compartmentalized and sorted differently. So we can have basolateral transported proteins and apical transported proteins. This is why influenza tends to stay in the lung tissue rather than spread. This is the paper I was supposed to talk about last Wednesday. Spending a few moments in Chapter 7, genome replication. Depends on what kind of a virus you are, so we go back to the classes and how you are replicated. So double-stranded DNA, of course, encodes its own daughter cells in the semi-conservative replication. Single-strand DNA, of course, would code for its complement, which could then be separated back out into the single strands. Double-stranded RNA can be replicated, as we'll see, in either semi-conservative or conservative ways. And then RNA that's there in the retroviruses is reverse transcribed into DNA before being transcribed back into the RNA for replication. So again, it depends on the type of virus, the host, the life cycle, as to how we do things. Replication must be primed. That can occur different ways. So normally, so for eukaryotic replication, there's a primase that synthesizes an RNA primer, binds to its complement, serves as a primer for DNA polymerase and replication. The hydroxyl group of viral proteins can also fill that function, as can a span at the 5' end of a DNA molecule that folds over and forms double-stranded sequence, giving what the DNA polymerase needs, of course, is not only some double-stranded sequence, except in the case of the protein, but a free hydroxyl from which to use the phosphotransfer from the triphosphate nucleotide coming in to form the phosphodiester bond. This is a replication fork. I have a last animation to show you here. The replication always occurs 5' to 3'. So as we have, in the case of double-strand DNA, we have helicase, which unwinds our DNA, and we have DNA polymerase, which extends the daughter molecule, semi-conservative, by the way, is of the daughter molecules, one strand is from the parent, one strand is newly synthesized. We have a leading strand where our polymerase synthesizes our new molecule in the direction the replication fork is happening. The lagging strand has discontinuous synthesis of new strands in the other direction. So we have synthesis, and then the synthesis will meet up with a previously synthesized fragment of DNA known as an Okazaki fragment. They have to be joined, new phosphodiester bonds formed to form one molecule. This is a misleading way of representing how replication occurs, because the polymerases don't move down the DNA. The DNA actually rather winds back up, and it's really cool. So let me pull up that animation to perhaps end the day.
DNA replication, both strands of the helix act as templates for the formation of new DNA molecules. Copying occurs in a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite 3' to 5' orientation, and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to and in some systems, multiple Okazaki fragments may be present.
Even this is a, well,
No DNA replication. As in,
so it's easy to see the leading strand here. You have this subunit of the polymerase. It's essentially taking the single strand that's coming out of the helicase and immediately copying, and you can very easily see that they're near to one another in space and remain. It's this discontinuity that replication fork is a great schematic, but it just shows you something that's a little bit out of character in that, excuse me, the lagging strand polymerase is actually also right near helicase, and it says synthesis occurs backwards. It's not really backwards. It's still moving in the 5' to 3' direction. It's that the strand is backwards relative to the leading strand, but that's the nature of antiparallel nucleic acids. I like this because it's more realistic, and that you get a whole, what's called the holo enzyme, so a multi-complex enzyme containing the helicase and 2 polymerase subunits, is always synthesizing in the 5' to 3' direction. What that 5', 3' direction is in space is different than the replication fork that's pictured not only in this textbook, but pretty much every textbook ever published. You know, they have a schematic like that. So that was double-stranded DNA replication. Again, a lot of the viruses, well, depends on the class of virus. So a double-stranded DNA virus is going to use DNA-dependent DNA polymerase. RNA to RNA is an RNA-dependent RNA polymerase. So forth. So on. So on and so forth. It's in the case of DNA to DNA and DNA to RNA that we can rely on host enzymes. Other ones have to be encoded by the virus. Let's go to place these things. I'll do chapter 7, 8, and the paper next time. So we're pretty up to date.